Hello and welcome to Biorules video lessons. Today we are going to be talking about microscopes. This is a short class because most of microscopes it's better when we learn by putting your hands on, playing with actual microscopes. Okay, so what are these microscopes and what do we need them for? Human eyes can tell things apart when they are up to 1/16th of a millimeter away. So if I ask you, for example, in your screen, how many dots do you see here? Unless you're very young or you are very, very close to the screen, you're probably going to say that you see one dot. But you're probably going, depending on how young you are and how good your vision is, start to see that actually there are two dots in this image here. But when they are very, very close, we don't tell the difference. So if you want to be able to see things that are very, very close together, so things that are very, very small, we need to magnify the image. We need to make the image bigger. So for that, we're going to be using lenses. So if you place two lenses, one in front of the other, and you adjust the focus distance, then you can make yourself a compound microscope. You are going to learn this in physics later. It's lots of fun. When you learn optics, you get the focal point of the lenses, you adjust them in the right distance, and you see you can potentialize the capacity of one lenses to magnify things. Usually, if you make it right, you can multiply the magnification of one lens by the magnification of another. So if you get two lenses that can magnify 10 times and you put them in the right placement, you can make a compound microscope that will be able to magnify things 100 times. Lots of fun for you to have in physics. Basically, this is a microscope. This is looks very complex, very fancy, but not more than one lens over there and another lens over here. You have two lenses, and then you put your sample to be able to observe. Just a little bit of a theory behind the structure of the microscope. So the microscope will have an eyepiece. Basically, it's a lens where you put your eyes, and it's going to have an objective lens. So it's the second set of lens. So you're going to put your eyes over here, and you're going to be seeing the light that will pass through the object here and get into your eye. This here is how we see the object. For that, you need a stage where you put the sample in a slide. You have some knobs to be able to move the stage sideways. And we have over here the focus knobs. You have the coarse focusing knobs and the fine focusing knobs. Here, usually they are both together. There's a further one and a closer one. And they move the tray here, the stage, up and down to try to put the object in focus. This here is called a transmission microscope because the light is transmitted through the material and then you're capable of seeing it with your eyes. Okay, here a few images for you. We have there liver cells. There you can see the cell, you can see the nucleus, and if you look very, very, very close, you may even be able to see some clamps inside the nucleus. That's a chromatin. So this here is magnified a lot for us to be able to see better these liver cells. And we can look closer here. If we use another kind of microscope, this colored one on the left is an optical transmission microscope. And this image here in black and white, they are using a different kind of microscope. They are seeing things even more details. This here is the nucleus that you saw before. This here, the pieces of chromatin with nucleus over there. And here we see even more structures inside of the cell because we are magnifying it even further. So here on the left, we have the pancreas seen in optical microscope. And here you have those same cells seen in an electron microscope that can magnify it even further. We see that this black and white image on the right has a much bigger magnification and resolution. We can see things much better. So this is the star of the show here now. Electron micrographs, unfortunately, they can only make black and white images, okay, but they have a much higher magnification. They also have a much higher resolution. Resolution is the capacity of telling two pieces of image apart. Basically, You know this by how many megapixels your camera has. Okay, so electron microscopes are amazing. They have a very big magnification, very big resolution, but they only see non-living material, because for you to prepare the material for electron microscopes, you need to spray a layer of metal. Usually they use gold, and that ultimately, if the material is already not dead, is going to be dead for you to be able to observe. Some images here we have these are images seen very, very much magnified. This is a mitochondria, a tiny little organelle inside of a cell. This here is inside a chloroplast, which is an organelle inside a cell, and this is part of a chloroplast. No, these are the reticuloids of a chloroplast. And this, the Golgi apparatus, also an organelle in a cell. So we are seeing things very, very magnified. They make amazing black and white images. Okay. The problem with electron microscopes, in addition to being black and white and only observing that material, is that it's much bulkier. Now, while you can have one optical microscope that's small, with the size of a small computer, and you put on your desk and you can watch it, an electron microscope can occupy an entire room. It's much, much, much bulkier, much bigger, and it's very, very, very expensive. Usually it's so expensive that only universities or other research departments are capable of buying them. Now, you cannot have an electron microscope in a school because they are very, very expensive. Okay, they are also kind of complicated to use. Usually in the universities or other research centers, there is a technician whose job is entirely to prepare and to make the images that are asked for by the researchers and the doctors and so on. So you need a technician just to play with the microscope. While the optical microscope, you put them in the top of your desk and you can use it yourself. Very easy. You are going to use optical microscopes in our classes here. Okay. So, light microscopes, living organisms, they can be seen. They can see images in color. They are relatively cheaper and they are relatively simple. So these are light microscope. Electron microscopes, on the other hand, they can only see that material. They only may see images in black and white. They have a high magnification resolution, but they are expensive, complicated, and bulky, and occupy lots of space. So this here is the difference between light and electron micrographs. Light and electron microscopes. Micrograph is the image. Okay. Transmission electron micrograph and scan electron micrograph, there are two different kinds of micrographs. The transmission is where the electrons passes through the sample. It's the same idea as the optical microscopes that are going to be seen in class. While the scan electron micrograph sees the surface of objects, is to see not through the cells, but seeing the surface of cells or the surface of small organisms like here, these ants. Okay, so those are two different ones. We are in biology, sometimes we may use some scan electron micrographs to see some structures, but to study cells, usually we want to see through the cell to be able to see the organelles inside. Okay? Just as a curiosity, there is a microscope called atomic force microscope that I am going to be talking about my experience using atomic force microscope when I was in college in Unicamp. When I was there, Unicamp had just bought the first one, and it's a microscope that can see things so small, so small, so small that it can see the impression of atoms. They actually don't see atoms because you cannot see an atom using something made of atoms, but they actually can feel atoms, and it's an amazing stuff. I can talk more about that later for you. Okay, I hope you had fun with this, and I hope you look forward for practical lesson when you are going to put your hands on microscopes to see some samples. Bye-bye.